everyone. For my project, I did the propagation of the wandering Jew, Transcientia fluensis. What is the wandering Jew? They are native to the tropical rainforests of South America, but they are introduced invasive pests in the southeastern of the United States, even in Australia and New Zealand. They are mostly grown in moist, rich habitats. They are described as a spiderwort, meaning they are weakly upright to scrambling plants. They have a perennial cycle. It considered as a houseplant that grown on pots as a hanger plant. Also, it considered as a garden plant as sprawling ground cover. Their outer edge of the leaf is striped dark green and silver with the inside purple in color. It is easier and faster to grow because there can be root formation in any node. There are a few journals on the propagation of the wandering Jew. In this journal article from Chester Soy, he states that propagation can be successful by using vegetative terminal shoot segments in water or a Hogan solution. This shows a successful root formation. Another journal article from David Kelly, he did a study on the effects of growth and regeneration on the wandering Jew. In his methods, he did stem cuttings that include nodes with and or without leaf in vermiculite at temperatures of 15 to 25 degrees Celsius and at different light intensities. In conclusion, it has been shown that high light intensity showed a better growth of the wandering Jew. According to this journal from University of Wisconsin, they used stem and leaf propagules in a matrix consisting of vermiculite, peat moss, bark ash, and wash sand at growth chamber conditions. They were watered with half-strength Hogan solution. The overall objective of this study was to compare the root growth in different types of media, including pro-mix soil, vermiculite, Hogan solution, and water. Another objective is to compare root growth with IBA, indole-3-butyric acid. From this website, I found the step-by-step methodology of how to take cuttings from this plant in order to root. And this website states the step-by-step methodology as the other one, however, it included the root synthetic auxin powder treatment. Here's the overview of the methodology using 4 different types of media. For each medium, the ontogenetically young and old cuttings were used with the control and IBA treatments. And here is the picture of the different media. Here's a video of how to propagate from the mother plant for the control. For the first step, the wandering Jew plant was placed in the water the day before the propagation process. For step 2, sterilize the scissors with rubbing alcohol. Then, cut the stem at the below level of the node. Since this is the control, the plant was not treated with IBA. Finally, place the cutting into the medium. For the results, plants in water had longer length and denser branching roots. There was some death of plants in the pro-mix soil, vermiculite, and Hoaglands, potentially due to many reasons, which I will explain later. Pro-mix soil with IBA treated had a new meristematic growth. Vermiculite had the shortest length of roots. Hoaglands had a control plant that didn't die but didn't form roots. The reasons why there was some death of plants in media was because plants were grown at my house with low light intensity, humidity, and temperature of 60 degrees Fahrenheit for 3 weeks. The plants that survived were transferred to ESF Green Campus Initiative's greenhouse and were grown for 2 weeks. There was higher light intensity, temperature of 70 degrees Fahrenheit, and humidity conditions, which would be more beneficial for better root formation. These plants were grown in water. As you can see, there was denser branching with IBA treatment. For the pro-mix soil, you can see a meristematic growth with the IBA treatment and denser roots. The vermiculite medium gave shorter root formation. However, you can see there was denser roots with IBA treatment than the control. For Hoagland solution, there was root formation with autogenetically young cutting, while there was none with autogenetically old cutting. Here's an overview graph. As you can see, plants in water have a positive root length and dense branching than the other media. The other media had many plant deaths. I searched on why watering Jew plants were dying, and I found these facts for their growing conditions. It seems that they grow better with high light intensity and average temperatures. For many possible reasons, plants were exposed to prolonged levels of low light and temperature of around 60 degrees Fahrenheit. As a result, they didn't grow well, and even some mortality was seen. Another reason was because the cuttings were taken too far below the node. As seen in this project, plants in water had the most positive root growth. There was more dense branching with IBA treatment, but there was no difference in root growth when comparing autogenetically young and older stems. Overall, we should not conclude that as a better medium than others. As a future study, I would like to repeat this experiment by having a larger sample size and letting them grow in greenhouse conditions. Also, I would like to test other media. Thank you for your time. Do you have any questions? Also, here are my references.